Hello, everyone, and we're very excited this week about this wonderful turnout. I'm even more excited, however, to have the privilege of welcoming Dr. Robert Williams to this week's NeuroTools Webinar series, as he introduces the NIDA Core Center of Excellence on Omics, System Genetics, and the Addictome. Dr. Williams is chair of the Department of Genetics, Genomics, and Informatics at the University of Tennessee Health Science Center. Dr. Williams holds the UT Oak Ridge National Laboratory Governor's Chair in Computational Genomics. He was a past president of the International Society for Behavioral and Neural Genetics and a founding director of the Complex Trait Community. He is also the editor-in-chief of Frontiers in Neurogenomics. One of Dr. Williams' more notable contributions is in the field of system genetics and expression genetics, EQTL analysis. He and his research group have built Gene Network, an online resource and suite of gene mapping code that is widely used by genetics and molecular biology communities. I will be adding these websites into the group chat if you would like to examine them. But without further ado, I want to welcome Dr. Williams as he introduces the NIDA P30 Core Center of Excellence in Omics, System Genetics, and the Addictome. I'm personally very excited to hear about how this core center will empower researchers to analyze genetic, epigenetic, and environmental causes of drug abuse risk, relapse, and treatment. So once again, thank you, Dr. Williams, for joining us today, and welcome.
My pleasure, Devin. Thanks for the very kind.
Sorry, Dr. Williams, I might have just muted you on accident. Hang on one moment. And you are back. Sorry, you might have to restart there. My apologies.
Yep. Just wanted to thank you for the kind introduction. And you mentioned, Devin, that if I felt compelled to, I should add a little bit to the introduction. I started out as a neuroscientist for all of you that are online. I have worked with the neuroinformatics framework from the get-go. I was the chair of the Society for Neurosciences Neuroinformatics Committee for a couple of years, and then Marianne Martone led the charge for a couple of years. Worked with Marianne and Mark Ellisman and many of you and many of the previous speakers here in the neuroscience field, but also in the neurogenetics field and genetics field in general. Over the last 10 years, we've branched out from neuroscience and instead of just doing systems neurogenetics, we're now doing systems genetics. We all know now that there's just so much crosstalk between systems, between gut and brain, between immune system and brain. And so it's really important to get tools that enable a holistic analysis to be practical. And I'll show you what we've been doing. The work I'm going to show you, and let me share my screen. The title is a little bit different than what is on the website, but not that different. Let me share and get this going. So systems neurogenetics using genetic reference cohorts and Gene Network. This project that I'm going to describe was initially funded as a human brain project starting in about 2000, 2001, at the same time that many other large projects were started. The Human Connectome project that you've heard about, some of the other work. I think Paul Pavlidis gave a talk a couple of weeks ago or months ago. That was also funded in part by this funding source, the Human Brain Project. Now, of course, there's a European Human Brain Project, but it's important to remember that the NIH, NSF, NASA project got started about a decade and a half before the European initiative, and some good things have come from it. NIF in particular, and Gene Network is the one of course I care about the most. It's really a good thing that we've been working in the trenches for the last 10 years. It turns out the tools we've developed are going to be very useful for various initiatives in the precision medicine area. What I'm going to argue in this presentation is that we need tools for experimental precision medicine, and I'll tell you what I mean about that. over the next few minutes. And by the way, I will not be able to see your questions if you type questions in, but Devin, I hope, will be monitoring those questions. And if you want to interrupt, get some clarity on a particular point, please do interrupt, and then Devin will I hope, flag your questions.
Absolutely. I will be monitoring the chat as well as muting anyone whose microphone is on. So if your microphone is on and you are not Dr. Williams, I do ask that you mute yourself. We do appreciate that, just so there's not too much static on our audio. Thank you.
Great. So this presentation over the next 40 minutes is really divided into two parts. I'm going to give a standard mini PowerPoint presentation. I think I have about 50 slides. And then I'll actually show you the web resources we've built. I'm going to do this in the context of drug abuse. We are funded by a new P30 that Devin introduced you to. At the same time, we have had funding for Gene Network, both from the Human Brain Project, from NIDA, from NIAAA, and now from NIGMS. And so I'm going to kind of mix them up a little bit, but I'll talk primarily about traits that are relevant to addiction and alcoholism over the next few minutes. The slide you're looking at, humans in the foreground, mice in the background. And the idea really is to figure out how to bridge between these two systems. As many of you probably know, there's quite a bit of controversy to what extent can mouse models inform precision medicine in humans in particular, psychiatric disease in particular, addiction in particular. And I have to say, if I go through my own, I've been bold. I even have a mouse models of dyslexia presentation somewhere. So I would argue that at some fundamental level, there's just a lot of commonality between mouse and human. That's true between Drosophila and human also. And we just have to look for that. We're really not specifically interested in gene variants that control disease risk. We're interested really in mechanisms and networks of molecules, whether they're proteins, metabolites, or mRNAs, or DNA molecules and epigenetic modifications. And at this sort of systems level analysis, the conservation will generally be better. And so there's definitely a place for animal models in as part of genome-wide association studies now, going from the GWAS hits to mechanisms. And there's definitely a place in early stage therapeutic testing. And the problem, I think, in general for mouse models is that most mouse models are on a single genetic background. And you'll notice in this slide, I've got at least two different types of mice. The one in the far background is C57 Black 6J. Kent Lloyd told you a little bit about the knockout project, probably, that the MMRRCs have been generating. All of those knockouts are on that guy in the background. Actually, that's the mother of our cross. And in the foreground, you see the two eyeballs, beady eyeballs of DBA2J, dilute brown agouti mouse. And we've made a family of these between these two parents. And I'll tell you about the family of mice we've been using for a lot of our work and how we think it's relevant for human neurogenetic conditions and diseases. Okay, so the key topics today, I've already mentioned a little bit of this, but what are we doing? So we're trying to facilitate the use of diverse genotypes of animals in neuroscience, whether they're rats or mice. That's our special expertise, those two species. Obviously, there's a lot of work in nematodes, C. elegans, danio, macaques, and other creatures, but my expertise and competence really is in rodent neurogenetics and rodent genetics. So I'll be talking about how we can help make it practical to study 10, even 100 different genotypes of mouse or rat. It's a huge burden to study one strain of mouse or rat adequately, and you can imagine how much more of a problem it becomes when study section or an NIH mandate insists that you study multiple genotypes to make sure that your results are robust, reliable, and generalizable. And so we're trying to make sure that that is actually something one can do with a standard R21, R01 budget. So how do we do that? How do we add rigor and enable more confident generalization from mouse, rat to human? So we're trying to do that. So that's goal number one, sort of the general goal. The specific goal is to actually implement this in a way that is useful today. And that's what I'll show you. I'll show you genenetwork.org. I see I'm missing a dot before the org there. And all of the tools I'm going to tell you about today, all of the software is open source. Most of the data is open source, unless it's new data that people have just deposited and are not comfortable with it, either for data quality concern reasons. It's been put in there as part of a proofing exercise, or it hasn't been published yet and they're worried about potential competition. But I would say that more than 90% of the data that I'll show you today is open. You can download it in bulk, you can download it in piece, or you can just use a system that we've built and analyze it within the web service in the same way that you can do quite a bit of analysis using the Human Connectome resources. I'm going to focus on mouse and rat. Whoops, sorry. But I will perhaps do a slight detour to the genotype tissue expression data set, the GTEx data that has been so well funded by all of NIH actually as a director's program. Quickly, what are the gaps? For those of you who have been in NIF for a long time or know about it, one of the challenges in neuroscience is what they call the long tail data in neuroscience. Most studies are done at the R01 and R21 level. You could make fun of that and call it cottage industry kind of neuroscience, and in some ways it is. It's really cool stuff. It's great to have control over questions and answers and research programs yourself, but it makes it much more challenging to integrate the data. And so that's referred to as the long tail of neuroscience data. And we're trying to capture more of that long tail data. Genomics data in general is not long tail data because it's usually well captured in anything from a FASTQ file to a geo submission. But there's a lot of very valuable phenotype data that we've been spending our time trying to capture. It's going into Gene Network. So Gene Network can either be thought of as a database, and it is a database, not a huge one, in the neighborhood right now of about 50 gigabytes. But what's really more important about Gene Network is the tier, the software application stack that sits on top of that allows gene mapping in real time. And you'll see what I mean by in real time and how real time is real time. It tends to be about a minute or two. And then how you can do some basic statistics and actually generate figures that are suitable for publications quite efficiently using Gene Network and the new P30. So the P30 is funded by NIDA. Its focus is in some ways similar to Gene Network itself. It'll rely on Gene Network. It'll also rely on the Phenogen project, a project led by Boris Tabakov and Laura Saba and Paula Hoffman at the University of Colorado, Denver. So it'll be hybridizing some tools. It'll probably be hybridizing another very cool tool called Gene Weaver that's run by Alyssa Chesler and colleagues at the Jackson Laboratory. But I'll be focusing primarily on Gene Network and mouse and rat data sets. What are the gaps? So here I have a list of some of the gaps. This will be obvious to many of you who hang out here at NIF. It's extremely difficult to assemble, curate, and make sense of all these data sets because, again, of this cottage industry approach. Maintaining code is a constant battle. There is no such thing as stable code, as far as I can tell, particularly if you're doing a web service. Developing suitable APIs of the sort that NIF is specialized in is also a constant battle. Those APIs change every couple of years. The standards change. I think for the good for the most part, but it means it's a constant struggle. And then, of course, the old metadata struggle. And this is still an acute problem when dealing with highly diverse phenotypes of the type that we encounter in the journal, the neuroscience literature. It's just very difficult to describe these. Even the method sections of papers are not usually systematic enough to really enable powerful searches of the type that NIF needs to be able to handle. And then finally, I mentioned this again, true precision medicine. This is a huge gap. We're going to be talking about precision medicine for many decades to come. We're going to be struggling to actually deliver it, except for very strong genetic and environmental effects. But we need to get those ROC curves. We need to improve the ROC curves. That means we're going to need to be able to do sophisticated modeling to do that in human populations of different ethnicities, different genotypes, different environments. We're going to really need great quantitative data. And I would argue that the efficient way to estimate the precision of precision medicine is going to be to use mice, rats, Drosophila C. elegans, and see how far we can get predicting phenotypes and gene by environmental action interactions and gene by drug interactions with these animal models. I think we can do that in the next 10 or 20 years. And I think we'll always be perfecting our predictive powers for human cohorts. Anyway, enough of that. So what I'm going to show you today is Gene Network, part of the P30 from NIDA. I will be showing you some of the data sets we have. And I should emphasize that the mouse and rat models that we're interested in using here are different from what many of you may have used or be using. A typical pattern is to use a single isogenic genotype of mouse or rat. In the case of mouse neuroscience, it's mostly C57 black 6. There are some users who use outbred mice, CD1, ICRs. Same thing in the rat neuroscience field. Some users use outbred Sprague-Dowley rats. Many use inbred, Fisher 344 or Wistar rats that have been inbred. There's some real benefits of using inbred strains, but there's some real risks. The obvious advantage is that for certain types of phenotypes, the variance is known to be non-genetic because you've got an isogenic set of creatures. The only thing that differs is you have males and females. So they're segregating for the Y chromosome, for the X chromosome, and for the mitochondria. But other than that, they're identical twins, essentially. The problem with that is if you study one genotype, that's an N of one study. No matter whether you study 100 or 1,000 Wistar rats, if they're all isogenic, if your black 6s are all isogenic, it's really an N of one study. And that makes generalization tough. So the idea is to study many isogenic strains of rat or mouse or Drosophila or C. elegans. The benefit is you can take genetically identical cohorts of genetically diverse, but still identical. I'm sort of talking out of both sides of my mouth there, but you want diversity but control of the genome. And with a host of different genomes, you can have the benefits of both. And you want to be able to put them in different environments and see how they do. You want to be able to age them. You want to be able to intervene early in development, late in development, and see what you can achieve. Here's where we're trying to go. This is the new name for the Precision Medicine Initiative. It's now referred to as All of Us. It used to be called the Precision Medicine Initiative. All of Us maybe is a little safer since it's not making any claims of precision right now. So I don't mind this change. But the idea really is to enable precise estimation of what the best intervention or prevention is or treatment is for you based upon your genotype and your environment. And that's going to be a pretty long haul. So here is, I don't know how many of you have followed the GWAS studies over the last few years. They come in for a lot of flack for a couple of reasons. I think it's unfair flack, and I think that the problem has largely been inadequate sample size. We are now doing way better in terms of sample size. So what I showed you in that previous slide here, the goal is to get up to 1 million subjects, normal subjects. So these aren't primarily disease subjects. And that's true for the Million Vet program also. So to get up to 1 million subjects, UK Biobank will have about 1 million subjects. With that kind of sample size, you can fish out very strong statistical results, and that's what I'm showing in this slide. So for opioid dependence now, we have the usual suspects, the OPRM, the mu-opioid receptors, the COMPs, the DRD2s, etc. But the sample sizes are getting so large now that we have now, I think, about 200 hits for schizophrenia. We have more than 250 hits for smoking cessation, for ever smoked, never smoked. The GWAS hits are coming fast and furious. You won't know it if you look in the literature right now. This is all recent stuff that was presented at a NIDA symposium a couple of weeks ago in Bethesda. But this work is really on the cusp of a very major breakthrough, in the same way that schizophrenia had its breakthrough in 2014 when the 108 schizophrenia candidate gene paper came out. So the problem is going to be, how do we go from GWAS hit, admittedly many with very small effect sizes, to networks of molecules and cells and circuits to potential treatments. And again, here's where I think the animal model work is going to be absolutely essential. And here I just flagged, for those of you who don't know about the NHGRI, European Bioinformatics Institute catalog, there's a lovely catalog where you can search for any term you're interested in and get a list of both studies and GWAS hits. So in this case, for this opioid dependence, you can see that there's a little button there that says 49 associations. Those will usually be very precise associations with very low effect sizes, but the effect size is a complete red herring. We don't really care whether the effect size is large or small. We can consider it an entry point. It's really a biochemical entry point into a process. And it really is the statistical p-value, an assurance that you've got the right SNP and the right gene rather than the actual effect size. So for those of you who track this, that's my 2 bits on this topic. So we're on the cusp of having an enormous number of GWAS hits, a lot of genetic success. But at the bottom of this side, you can see what is the immediate impact on patient treatment, and that is a problem still. So mouse resources, rat resources to the rescue. I'm taking you through a standard rodent pedigree here to make a permanent resource of identical twins, essentially. And so at the top of this page, you can see a mother and a father. I've shown a pair of autosomes there, color-coded in green and red for the 2 strains I showed you on the first slide. Those are the classic inbred strains. So the C57 Black 6, canonical mouse, all of the sequencing is done, all of the epigenetics is done with that particular strain, but it's just one strain. DBA2J, another strain. It's the first inbred strain of mouse, inbred in about 1909. You can make the F1 intercross here, sort of in the middle, the first row down. This is stuff you had in high school or college genetics. You'll notice that every mouse here has the same genome, 1 green, 1 red chromosome, so they're isogenic, but they're not inbred anymore. So they have allelic differences. So that is the F1 generation. The F2 generation is what Mendel used and what most neurogeneticists have used. The problem is every animal here is genetically unique. So that means you can't do highly effective gene by environmental interaction studies because you can't put the same genome in multiple environments. You can't easily do a gene by development study on a particular genotype, again, because every animal is genetically unique. So the simple trick here is to throw a male and a female F2 in a cage and inbreed for 20 generations their progeny. And you end up with a new inbred strain of mouse. So here's strain 1 of this B by D family. So the mother is the black, father is dilute, so B by D family. And we now have generated, with support from NIH and NIDA and NIAAA, about 150 of these strains. And the same thing has been done for rats. So there's a rat family of these so-called recombinant inbred strains. There are currently about 30 of them, and we're expanding that with help from NIDA and the director's office. We hope to have about 96 different inbred rat strains that can be used for experimental precision medicine. And the key thing about the bottom is just to note that we've got a set of many different strains of mice, 152 here. They all come from the same family. So we have the advantage of very powerful genetics because we have a family rather than a GWAS. We don't have quite as much precision as a GWAS, but good power. And we have the ability to come back to any particular strain of mouse or rat and redo an analysis to see how reliable our results are and to see whether we can intervene in a smart way, say to prevent glaucoma. So these mice do segregate for glaucoma. This father strain here has glaucoma. It has cardiomyopathies. It's highly sensitive to stress. This strain over here, the C57, tends to be a heavy drinker mouse, by mouse standards anyway, whereas this strain doesn't drink. There are a huge number of drug addiction differences. There are differences in impulsivity, et cetera. Any questions at that point? If not,
I think you're clear. I'm monitoring the chat. I haven't seen any questions come through yet. I think everyone's following along.
All right. Well, thanks, Devin. So you should have a kind of good take on the resources, the mouse and rat resources, this ability to resample a genome, also shown in this slide with 2 of each. But again, you can have hundreds of each. Each one of these mice or rats can cost anywhere from $20 to $200, which is an issue. So one of the problems is to make sure that all of the data that we generate gets combined, and that's what Gene Network is doing. The slide over here just shows that we can do both forward genetics. We can start with a trait and ask what gene variants control that trait. So that could be impulsivity, for example, or response to cocaine. Or we can actually start with a gene variant, and we can say, does this gene variant control trait X? And that's called, in this slide, it's called reverse complex trait analysis. It's now also called phenome-wide association study, where you have a phenome and you ask, does gene X have effects on this phenotype, that phenotype, or the other? So you're actually scanning across the phenome rather than scanning across the genome. This is an example of this kind of reverse complex trait analysis or phenome-wide association study. In this case, the well-known gene that degrades a lot of the catecholamines, catecholamine methyltransferase, is in the middle of this circular diagram. There's a known variant in the 3' untranslated region of this gene that results in about a 2-fold difference in expression between 2 types of mice. They actually are the 2 types of mice that are the parents of the BXD family I just showed you about. And then you can ask, what does the impact of this insertion have on the expression of genes in nucleus accumbens, in hippocampus, in prefrontal cortex, in striatum? And you can see here, for example, that monoamine oxidase is controlled by catechol-O-methyltransferase. And we know that that's a causal relationship because we can do the genetic mapping. And the genetic mapping says that the variation in this gene is directly causal to the difference in monoamine oxidase expression. We can also go up a layer and look at protein levels. So here we're looking at DRD one, protein density. We can look at neuropharmacological responses to drugs such as haloperidol, etc. So this is a fairly new method. What I'm trying to leave you with is that if you have a large phenotype collection for a family of mice or rats, you can do both classic forward genetics, going from trait to gene variants, but you can also flip it around and go from gene variant to many phenotypes if you have good data. And that's why this long tail data problem needs to be taken care of. We need to collect as much data as we can for our family. And by the way, in this figure, I've got these little blue and red arrows. Those are simply correlation coefficients, in this case, between the genotype at COMPT and the expression level of the genes or the response to pharmacological agents like haloperidol. I'm going to skip this slide in the interest of simplicity, but it really just says that you need to titrate the complexity of your problem. You can either have a very reduced system, which is quite simple to handle, but may not generalize well to human populations or to other mice or rats. And so it's sort of a Goldilocks problem. How do you get the right level of complexity in your system? The BXDs are pretty complex. So I just mentioned one mother, one father, but they're segregating for 5 million sequence variants, about 10,000 missense variants. So that's about as much as you'd find in fairly large human cohorts, all of Iceland, all of Finland. So it's one family, but it's sufficiently complex to be challenging for us at this stage of analysis. This is what a phenome looks like. So at the top again, strains of mice, and then we have different levels of phenotype. At the top, we have classic phenotype. So something like impulsivity would be a classic phenotype. We have about 5,000 of these classic phenotypes. Almost all of the data is quantitative. So if you want to look at David Jensch's impulsivity data, it's in there as a quantitative phenotype. In this case, it would be the number of trials to reversal criterion in a touchscreen assay. Below that, you'll see protein and metabolite data. Every bar here, by the way, is a strain of mouse from BXD1 to BXD about 102. You'll see some gaps here. So there's not, in this protein and metabolite, there's a gap here, and that's simply because the group that generated all of this cutting edge protein and metabolite data focused on the high numbered strains. And then below, and this, by the way, is 40,000 traits. So good, but not great. And then for the mRNA data we're in the millions here. So this is 3 million traits. So we have typically more than 1 million mRNA assays, mainly array, but more and more RNA-seq data. So that's what a phenome looks like. And the question is, what kind of phenomes do we have for human populations? And the answer is, we don't really have quantitative phenomes for any humans. So the best we're going to do over the next few years will be the All of Us program, Biobank. Most of the data, though, tends to be qualitative. Has disease X, does not have disease Y. Okay, so now we're getting ready to go live. And I actually will shift live. I have a presentation, so for those of you who want to download the rest of the presentation, it'll give you a static version of what I hope we'll do right now. So I'm just going to escape from PowerPoint and then just go live. So Devin, I hope that appears pretty well.
Yep, it looks crystal clear on our end.
Okay, so this is what the production version of Gene Network looks like right now. You'll notice that it says use Gene Network 2. It should say use Gene Network 2 at your own risk. It's much better code, so if you're interested in working with any of our team on coding or user interface, Gene Network 2 is our state-of-the-art. I don't know if it's the state-of-the-art, but it's the best we can do. It's all available on GitHub. I provided that link in the second slide. I won't show it just because it's a little bit too alpha right now. So it's not quite as fast as we want it to be. But it does basically everything we're going to do in this Gene Network 1 demo. Just for those of you who are NIF, this is registered in NIF, as you can see over here. And this has been going for a long time. I mentioned it's really been going since about 2001, but we had an earlier website called the Portable Dictionary of the Mouse Genome. And those are some of the people that have contributed to it. Here's some of the more sophisticated search terms. A lot of you on NIF will be comfortable with more complicated searches. Simple structure, relatively simple, maybe not simple enough, but you choose your species. The human GTEx data that I mentioned is here. We have many other human data sets. You've had presentations on the Human Connectome project. We have some of the data there, but we don't have genotypes yet for the Human Connectome. You'll notice that these first two up here are with genotypes, and that allows you to extract causality from the genetics rather than just looking at correlation matrices. Here's another one that we're doing with genotypes. Human data is good, but I'm going to focus on mouse and rat because I want to keep the topic here on kind of experimental precision medicine. Mouse and MM10 is just the assembly that we're using, so Mus musculus version 10. This is rat norvegicus version 6, and I will take you here to the BXD, and then I'll show you what I was showing you in the previous slides, phenotypes. So I could just put a wildcard in here, and it would take a few minutes to download everything, but there are about 5,000 traits that I mentioned in there. But let me do the search that I just showed you, which was this one. Cocaine, nicotine, opioid, methamphetamine, alcohol, cannabinoids, and do that search. So that's now happening. And I got 585 records. Basically, you can think of each record as a vector of numbers. And I'll show you one of these. So let's just look at this first one from a 2016 study. Let me just click on it, and you'll see what these vectors of data look like. So this is where the metadata becomes a hassle. We have to define our own phenotypes. So we've got a little sort of mini ontology going here. Central nervous system behavior pharmacology, that's our controlled vocabulary, very simple. Then a description that we've gotten from the authors usually, and I often have to beat them over the head several times to get these. Then if it's from a figure in the paper, we may have that, and then the units in square brackets, linked to PubMed. And what does this vector look like? Well, here it is. These are all the individuals that have been studied. If it's X, that means there's no data for that particular member of the family. The yellow here means it's an outlier. So we're flagging it for the community, and it goes on and on. And you might, seems like a dumb way to display the data, but we do it that way because you can actually edit it. Here's the data in another format. 29 different strains have been studied. Here's the QQ plot. If it were normal distribution, it'd be a straight line. Here is the data as a bar chart showing you the different strains. That's BXD1, BXD9, BXD100. We can sort it this way and just look at it in rank order, essentially. Again, very simple stuff, but you can see you can just pull these off from your desktop, and they're almost ready for prime time and publications. So a lot of users just yank those off, and we're trying to do it a little better. You notice that was a PNG or a GIF. We'd like to do this in vector graph. But what we can do that's maybe more fun is I can ask, what is the correlation between this particular trait and this is cocaine self-administration. And I might want to know, is there a correlation between that behavioral trait and gene expression in midbrain? So these are all very large genomic data sets, and I'm heading down for midbrain here. Again, the interface could maybe be a little, it's grown beyond our capacity, but now I'm asking a question. How does this trait here, self-administration, co-vary with gene expression in the midbrain? And I'll just click that. And I haven't tweaked the original vector of data. You might want to do that. So this is why the probability plot suggests we have some high outliers here. If I did the box, you can see I have 2 outliers that I should perhaps have taken care of. And that's why this is useful. I can sort these, and I can say, oh, I have 2 outliers. I could either delete them from the analysis, or I could trim them. I could Windsorize them. I could redo it. So the important thing is to allow the user to actually work with the data so that they don't have to download it and do it in R or MATLAB. So I computed the correlation between that vector of data and all other transcripts, or all transcripts in the midbrain from an Agilent microarray experiment. The correlations are right here. Some of them are positive, some of them are negative. And I can look at these. I could select them all. I have 1,000 covariates here. I could export this. So I go to the top and I could say, just export that list. I want to do something clever myself, download the table. I could say, I just want the negative covariates, please. Show me how many negative covariates you have there. And it will select just the negative covariates. I could say, just select the negative covariates that have reasonably high expression. Let's call that 7. Select those. So now I've trimmed it a little bit, and then I could do a classic gene ontology analysis. So I don't know how many traits here in yellow, let's call it maybe 100, but I can do a gene set analysis of the negative covariates with high expression in midbrain of my cocaine response. And now I'm sending that data set to Vanderbilt, 141 mRNA assays, 141 vectors of data are going to Vanderbilt. Actually, I think this is now at Baylor. And they're running a gene set analysis for us. So this is sort of the family of web services that all of you in NIF are very familiar with. And we have a small group of web services we work with. So now I had 148 transcripts that covaried, and I can see how they're enriched here in a gene ontology for molecular function. So this is not a killer analysis, but you can see that we have 3 genes that are supposedly in this particular category. Active transmembrane transporter activity is enriched. With only 3 genes, I would take that with a very large grain of salt, and I would consider that analysis pretty much a negative result. I could flip it around though quite easily, like that. I'll have a different set now with positive correlations, and I could redo it on the positive side. But that gives you an idea of how we can, so what are we doing here? So I mentioned we've got up here at the very top, we had all these phenotypes. And then the reason I can compute a correlation is because all of these strains of mice have now been used for over 40 years by many investigators. In this case, Randy Blakely generated these data at Vanderbilt. He generated mRNA expression data using the Agilent system on midbrain samples from the same strains, obviously not the same animals, but the same genotypes. And I can add I can now build up a network of traits. So I just added the cocaine data into my BXD trait collection, and we could have a rat collection or a human collection. That's the trait I just added. I already had some traits in my collection from earlier this morning from some work I was doing. I might want to flip this and just remove the extraneous traits. Now I just have one item in my collection. But let me go to another data set. So I'm going back to the root level. And I realize we should be wrapping it up and letting you guys ask questions now. But I want to show you one final thing before I wrap it up. I've shown you how to look at a vector of data, how to search for it. You just search your term here. But what I haven't shown you is how do you get beyond correlation to causality. And let me give you a good example let's have a look at catechol O-methyltransferase that I mentioned before. So I'm going to look at the expression level of catechol O-methyltransferase in hippocampus. And I mentioned to you in my third or fourth slide that we can take this gene, a key gene in dopamine and catabolism. Let's actually look at the gene expression level. So in this case, our phenotype is small or big differences in gene expression level. In this case, the hippocampus. So again, here's my vector of data with some missing data and quite a few strains. Let's look at it at this level. 99 strains were studied. It is anything but normal. So this is what a Mendelian trait looks like. It's got a high group and a low group and very few in between. That is a wonderful distribution. It looks a little funky and risky, but it's actually what a geneticist wants to see if they want to do Mendelian genetics. If we sort it this way, you can see again which strains are low and which strains are high. And the question in your mind should be, where is this variability in catechol O-methyltransferase coming from? I gave away the story. I told you that it's coming from the gene itself, that there's an insertion in the 3' UTR. So let's see what that looks like if we use this mapping tool. So I'm going to use the mapping tool. Normally, in the bad old days, in the late 90s, the analysis I'm doing right now would have taken about 3 hours. So what we're doing now is we're mapping the trait being catechol O-methyltransferase expression in hippocampus. We mapped it with 1,000 permutations. This is chromosome 1, chromosome 2, chromosome 3, going off to chromosome X on the X-axis. And then on the Y-axis is the genetic mapping LOD score or likelihood ratio statistic. So this is our statistical confidence that we have causality. And here, obviously, we have something big happening on chromosome 16. That happens to be precisely the location of catechol O-methyltransferase. So really, we're saying something very simple here. We're saying that differences in catechol O-methyltransferase expression are caused by a gene variant in or very close to the catechol O-methyltransferase gene itself. Where did that variation come from? It came from an insertion in the 3' UTR, as I showed you earlier, a paper we published in PLOS 1 quite some time ago. So here's what that map looks like. It's a little uglier now, but this is one chromosome, chromosome 16. All of these little marks here towards the top are the genes there. There are a total of, let me scroll all the way down here. There are a total of about 1,200 genes on chromosome 16. And some of them are in regions that are highly polymorphic. So all of this orange hash you see here are regions where the two parents, the B and the D parent, differ a great deal genetically. And then some regions of the genome are very similar between the parents, the B and the D parent. And you'll see that's the case here. So even though catechol methyltransferases in a region that's almost identical by descent. In fact, it's not identical by descent because we know we have that retrotransposon that got plunked into the middle of the 3' UTR in the mother of the cross, the C57 black 6. So this is how you get causality. And now the second question is, what traits of neuroscience interest in the BXDs specifically map into this chunk of chromosome 16? And that's what we referred to as the phenome-wide association study that I mentioned earlier. So I'm going to wrap it up now, but what you know is basic statistics, calculate correlations, mapping tools. I can add this into my shopping cart. And now I have two things in my shopping cart: catechol methyltransferase mRNA expression level, cocaine. I could ask a very simple question: do those two covary? The answer is no. In this particular case, they do not care for each other very much. So here we've got the low catechol methyltransferase group, the high group, and you can see that the expression of this behavioral trait or pharmacological trait does not differ significantly. P-values up here. It's getting interesting, and maybe if we had a couple 100 mice, it would be significant, but it's quite weak. So you can test a specific hypothesis that way. You can generate hypotheses, and there are a whole bunch of tools that I don't have enough time to show you that are here that do network analysis. For those of you who want to go a little deeper, just click on this reference and have a look at this paper. So Gene Network, a toolbox for systems genetics. If you just click here, you'll get a PDF, and it's divided into two parts. It's basically a how-to, a primer. It's a protocols article. Half of it is devoted to rodent models, half of it is devoted to human models. So it's pretty much just walk through it, do this, do that. So, you know, very, very granular how to do it. So that should, if you have any specific questions, and I'd be delighted if the one category of email I do routinely answer quickly is any problems or in using Gene Network or the P30 support. Anything you have related to drug abuse, delighted to help. But basically any problem where you think you can use our tools, I'd be delighted to help. Devin, I'm going to stop right there and open it up to questions.
Thank you so much, Rob. I always enjoy hearing about how the tools are evolving over time. And I know that so now this is Anita, of course.
Yeah,
and,
hi Anita. Good to hear you.
What you're, I want to ask you about something that you didn't really broach, but you kind of alluded to it, or you actually even showed it, that, you were registered with NIF, and
right.
We now have these resource identification numbers that I'm going to have to bug you about.
Okay. Alyssa's bugging me about it too. So I'm going to be working with Alyssa to take all of our phenotypes and give them phenome database identifiers.
Oh, that's a great idea.
We'll probably start with the BXDs and then move on to the rats, but I'd be delighted to work with you. And are you working with Alyssa Chesler routinely, or Molly Bogue?
I would say semi-routinely, but
okay.
Yeah, I mean, they just put out that really nice new paper we're trying to get them to talk about it here in this series, and so hopefully in the next couple of weeks we'll hear all about that aspect of it as well. But the just the resource identifiers are a really nice way to be able to track the use of your tools inside
right.
of the literature. So this is something that, I'm just going to come back and bug you about.
Please do. You respond to whoever's bugging you the most.
Yes.
and right now you may know Abraham Palmer, who's who handles research in psychiatry at UCSD, but I've been working very closely with him, and he's been pushing us on certain algorithms. But I think that for the big user community, actually getting the data we have organized better and more searchable and linked to NIF would be really critical. And getting NIF, Gene Network, and Gene Weaver and Phenome Project all together would be marvelous.
Yeah, so. and then I have two other kind of also more resource related questions. So I've been heartened over the last few years to really do, see a lot of the increase in the genotyping and the full scale genotyping of so many critters and of course humans, to where that data is now finally starting to come up with some significant, reasonable kinds of answers. And so I think it's always really interesting to see a field where at first there's all this excitement, but the data is really bad, right?
Yeah.
And then all of, everybody's kind of let down because the data was really bad and they finally figured this out. And, but then, you actually do start getting enough data to where you really should be excited. So it's sort of like, to me, it's this opposite graph, right? It's a new technique, so it's shiny. Everybody likes to use it. They overpromise, underdeliver. But then when the data is actually there, people are like, oh yeah, that again. But I love seeing the fact now that a lot of these things, the ends are large enough where you can actually start to track down some of these things. And I know you really did address that, but I would just like to highlight, it's now the genomics era, and we're at the point where we have enough genomes to start to pull out these probably very small but highly significant things.
Yep.
And we really did have, one of the biggest, absolute biggest, loci that ever came out, and it was an addiction. It was in addiction to cigarettes, and it's just, hey, this is a great win, but it wouldn't have happened with 10 studies, 10 humans, right?
Yeah. What frustrates me is that the leadership at NIH often is out of sync with where things are going to be in 2 or 3 years. So the Wayne Gretzky motto of skating where it's going to be is really important. And I think the leadership is often looking for the immediate win. And GWAS has been promissory, but I think it's finally there. And I think the next year will prove that for just about every addiction where the sample sizes get above 50,000. That really is sort of the critical threshold. Below 50,000, you kind of get the usual suspects, things like CHRNA5 for smoking. But now with 250,000, they're getting lots of new surprising hits that are real. They're not big effects, like you say, but who cares?
Yeah, no, who cares? It doesn't matter if it's important for that small population, that small percentage of the population. Then
or it's just an entry point. It just gives you something to grab onto, and you can try to figure out, well, maybe it's not polymorphic because it's so damn important that humans all have the same allele except for a handful. But that doesn't mean it's not a very important target for a potential drug intervention. So it doesn't have to be polymorphic to be druggable.
Absolutely. Okay, I'm not sure if there are any other questions. I don't really see anything unless it came privately to Devin.
No, I think we're all clear as far as questions go.
I think we're out of time one way or the other. So I think Devin's going to wrap this up and tell us how to get a hold of Rob if somebody wants to really ask a lot of other questions about
absolutely
this tool and other tools, and of course, which are the best targets to go after with my.
I'd be delighted to do homework for anybody online. As a matter of fact, it's my obligation. If you're funded by NIDA or NIAAA or NIGMS, I'm yours.
Awesome.
And I'd just like to add that this information will be in the YouTube description in a couple hours when we put this on our YouTube channel. You'll be able to access this video through a link I've just placed in the chat, newinfo.org/about/upcoming webinars. And I'll also contact you, Dr. Williams, in regards to putting that link to that published paper that you featured as well.
Absolutely.
We could add that in the YouTube section as well. I think that'd
That'd
be
be
highly
terrific.
relevant.
And I'll put your YouTube link on Gene Network. So we'll
right on.
Excellent.
I love it. Personally, I'd just like to thank you. This was an excellent webinar. I'm looking forward to putting this on YouTube. And if there's anything else you'd like to add at this point, please feel free. Otherwise, I think I'll end the recording and call it in on this.
And thank you very much, Devin and Anita, for doing this.
Great. Thank you very much, Dr. Williams. And I'd also like to add that we expect our next webinar to be on February 16th, but we are also in the process of adding on new webinars. We're in correspondence with a few researchers and other presenters right now. So we're excited to see where this webinar series goes.
Terrific. All right.
Great. Well, thank you, everyone, and I hope you have a wonderful weekend. I'll be ending the recording and ending the webinar right now.
All right. Adios.
All right. Thank you, Dr. Williams.